recombinant DNA technology. Now, this recombinant DNA technology, also known as genetic engineering, is a set of techniques which enable us to isolate, identify and recombine DNA from various sources so that we can introduce new characteristics in an organism. In genetic engineering or recombinant DNA technology, the DNA from different sources is identified, isolated and recombined, and this leads to introduction of new characteristics in an organism. That is what the genetic engineering or recombinant DNA technology is all about. Now, the invention of recombinant DNA technology, which involves the artificial introduction of genetic material of an organism into the genome of another organism, and that another organism then replicates and express that artificially introduced genetic material. All of this is largely contributed by the work of Paul Berg, Herbert W. Boyer and Stanley N. Coher. These three scientists invented all this. It was Paul Berg who first developed the recombinant DNA molecule, and this recombinant DNA molecule that was developed by Paul Berg combined the DNA of SV40 virus and lambda phage. DNA from this SV40 virus and lambda phage were combined, and that produced the recombinant DNA molecule, the first recombinant DNA molecule, which was developed by Paul Berg. After that, Herbert Boyer and Stanley Cohen developed the recombinant DNA technology, and these two scientists showed that the genetically engineered DNA molecules can be cloned in foreign cells. Genetically engineered DNA molecules can be cloned in foreign cells. This was shown by these two scientists, and that is how the development of the recombinant DNA technology took place. One important aspect of recombinant DNA technology is DNA cloning. Now, this DNA cloning is a set of techniques which enables us to assemble recombinant DNA molecules and to direct their replication in the host organism. With the help of DNA cloning, with the help of this technique, we can assemble a recombinant DNA molecule, and then we can replicate that recombinant DNA molecule into our host organism. Now, this DNA cloning, this term has this word cloning. Now, this cloning word indicates that the methods involved in this process consist of replication of single DNA molecule starting from a single living cell, and then this generates a large population of cells with identical DNA molecules. So from a single cell, lots of cells are produced and they all are identical, and that is the reason why the term cloning is there in DNA cloning. DNA cloning. Now, this DNA cloning is the production of large number of identical DNA molecules from a single ancestral DNA. You take a single ancestral DNA, you can call it ancestral, and then with the help of that you produce a large number of identical DNA molecules, and that is what DNA cloning is. Now, the essential characteristic of this DNA cloning is that the desired DNA fragment is selectively amplified, and this results in the large increase of the copy number of the selected DNA sequence. The desired DNA fragment is selectively amplified during DNA cloning. Now, in practice, the DNA cloning involves multiple rounds of DNA replication, and this DNA replication is catalyzed by DNA polymerase, and this DNA polymerase may act on one or more type of the template DNA molecule. Now, this DNA cloning involves two approaches. There are two approaches by which you can do DNA cloning. The first approach is cell-based, is known as cell-based DNA cloning, and the second approach is cell-free DNA cloning. Now, let's talk about cell-based DNA cloning. Now, this cell-based DNA cloning was the first form of DNA cloning to be developed, and it is an in vivo cloning method. Okay, this cell-based DNA cloning is in vivo cloning method. Now, the first step in this cell-based DNA cloning is attaching the foreign DNA fragments in vitro to the DNA sequences that are capable of independent replication. Okay, you take DNA sequences that are capable of independent replication. Then you take the foreign DNA fragment that you want to clone. Okay, and then you attach that foreign DNA fragment to the DNA sequence that is capable of independent replication, and this you do in vitro. Now, when this foreign DNA fragment attaches with this DNA sequence that is capable of independent replication, the product that you get by attaching these two is called recombinant DNA fragment. And this recombinant DNA fragment is then transferred into a suitable host cell where it is propagated selectively. Okay, this is how the cell-based DNA cloning works. You produce a recombinant DNA fragment first by attaching the DNA fragment of interest with that DNA sequence which is capable of independent replication, and then you transfer this recombinant DNA fragment into a suitable host cell where it is propagated selectively. Now let's discuss about the steps of the cell-based DNA cloning. Now, the first step is the construction of the recombinant DNA molecules. This recombinant DNA molecules are hybrid DNA molecules which consist of autonomously replicating DNA sequence plus inserted element. Okay, these two combines to make a hybrid DNA molecule which we call as the recombinant DNA molecules or recombinants. Now, such hybrid molecules are also known as chimera. These recombinant DNA molecules are constructed by in vitro covalent attachment, which is also known as ligation, between the desired DNA fragment called as target DNA and a replicon, which is any sequence which is capable of showing independent replication. So the ligation between the desired DNA fragment and replicon gives you the recombinant DNA molecule. Once again, replicon is any sequence which is capable of showing independent replication, and the desired DNA fragment is your target DNA that you have to clone. When these two are attached covalently in vitro, then that gives you recombinant DNA molecule. Now this step, okay, this attachment of the desired DNA fragment with the replicon is facilitated by cutting the target DNA and the replicon molecules with a specific restriction endonucleases. And then there is joining of these different DNA fragments with the enzyme DNA ligase. Okay, specific restriction endonucleases are used to cut the target DNA and replicon molecules, and after that they are joined by the enzyme DNA ligase. The next step in the cell-based DNA cloning is transformation. Now, during transformation, the recombinant DNA molecules are transferred to the host cells, such as bacterial cells or yeast cells. And in these host cells, the chosen replicon undergoes DNA replication independently that of host cell chromosomes. Okay. The recombinant DNA molecule which is transferred to the host cell, the replicon of that recombinant DNA molecule undergoes DNA replication, and this DNA replication of the replicon is independent, that is, it does not depend on the replication of the chromosomes present in the host cell. After transformation, there is selective propagation of cell clones. This is the next step. Now, this selective propagation of cell clones involves two stages. First, the transformed cells are plated out by spreading on agar surface to encourage the growth of well-separated cell colonies. Initially, what is done? Well, the transformed cells are plated out by spreading on agar surface, and this encourages the growth of well-separated cell colonies. Now, these well-separated cell colonies are known as cell clones, okay, because they are population of identical cells which have descended from a single cell. Okay, so by spreading on agar surface, the well-separated cell colonies that you get are known as cell clones because they are the population of identical cells that have descended from a single cell. In the next stage, the individual colonies are picked up from the plate, and those individual colonies are then further expanded in the liquid culture. After selective propagation of cell clones, there is isolation of the recombinant DNA clones, and that is the last step of cell DNA cloning, cell-based DNA cloning. Now, this isolation of recombinant DNA clone is done by harvesting the expanded cell cultures and then selectively isolating the recombinant DNA. This is how you do the isolation of recombinant DNA clones. So this is how the process goes of cell-dependent DNA cloning. You start with a cloning vector, you take DNA, you combine them and make a recombinant DNA. Now in the process, there is formation of recombinant DNA, but there is also formation of non-recombinant DNA. And you don't know because right now you can't see the DNA. DNA is very small. After this, you introduce these recombinant and recombinant DNA, which you don't know which one is which in the host cell. Okay, you introduce this DNA molecules into the host cells, for example, bacteria, And after the experiment, in the suspension, you have transformed cells which has the recombinant DNA. You have the transformed cells which has the cloning vector, but that is not recombinant. And you have non-transformed cells, that is the DNA didn't enter the cell. So these the three types of cells are there in your suspension, and this is bacterial chromosome which is represented here. Now after this, in the suspension, again you can't see which cell got transformed, which cell did not got transformed and all, because of obviously the cells are also small and you cannot directly see the DNA, recombinant DNA in them. So you take that suspension and you do selection of transformed cell. That is, you put something in the media so that the only transformed cells are capable to grow. And after that you get this transformed cells in your culture. Now both the cells are transformed. Okay, there are two types of cells in your culture. Both of them are transformed, but the one contain recombinant DNA and the one contain normal DNA, that is non-recombinant DNA. Now your motto is to isolate this recombinant DNA cell, the cell which contains this recombinant DNA. So that is the reason why you do again selection of this transformed cell with recombinant by growing these two cells on a medium which only allows the growth of this transformed cells which has recombinant in it. Okay, and after that you get the cell which has the recombinant DNA in it, and after that you amplify that cell, you get many cells, and that is how your target DNA molecule is now amplified. Okay, it is cloned into many. So this is how the cell-based cloning is done. Thank you. Let's also discuss about the cell-free DNA cloning, that you don't depend on the cell. The cell is not involved in the DNA cloning, that type of DNA cloning. Now, polymerase chain reaction is the newer form of DNA cloning, which is independent of the cell. And this polymerase chain reaction is enzyme mediated and it is conducted entirely in vitro. This PCR, that is polymerase chain reaction, was developed by Kary Mullis. PCR is a revolutionary technique which is used for selective amplification of the target DNA sequence by using short primers. Okay, in PCR you use short primers and you selectively amplify the target DNA sequence. Now this PCR is a rapid, inexpensive and simple method for copying the specific DNA sequence. It is rapid, it is inexpensive and it is simple.